Today's episode, Natalie and I interviewed Dr. Seyfried, an American professor of biology, genetics, and biochemistry at Boston College, who's been published in over 150 peer-reviewed publications. He received his Ph.D. from the University of Illinois in 1976. His postdoctoral fellowship studies were in the Department of Neurology at the Yale University School of Medicine, where he served as an assistant professor in neurology. His research focuses on mechanisms of chronic disease such as cancer, epilepsy, neurodegenerative, lipid storage diseases, and caloric restriction diets. So without further ado, here's our interview with Professor Seyfried.
Hold up, one more thing. If you have not yet scheduled your 45-minute free exploration call to find out what the root cause of your symptoms are and your next steps to getting symptom free, you are going to want to pause this podcast and go do that right now. So I invite you to go to nutritionwithconfidence.com, book your free exploration call, and find out how to get your results and what is possible for you.
Well, welcome, Professor Seyfried. Appreciate you taking the time to be on the podcast today. I wanted to start off with, I think most of us have grown up with this understanding of cancer development as largely being based in genetics. You just get just luck of the draw, bad genes. Um, so I wanted to start with, could you describe the origins of this theory as cancer being genetically based?
Yeah, well, thank you very much. It's nice to be here today. Yeah, that is the key. That is the key question that has to be addressed. You have to realize that in science, we move forward with our understanding of nature by developing theories to explain facts. So facts are collected, facts are analyzed, and then they're interpreted under a particular theory. And that theory can persist for long periods of time, or it can be overthrown by new information that explains the facts in a different way. You know, one of the ways that we look at these things is we look at the heliocentric versus the geocentric theory of the solar system. And for 1,800 years, from the time of Aristotle through Claudius Ptolemy, astronomers thought that the Earth was immovable, and sun, moon, and all the planets revolved around the immovable stationary Earth. And then it was the work of Copernicus, Kepler, and Galileo that provided new information that if you rearrange some of the celestial bodies, putting the sun in the center and the Earth as simply another planet, a lot of the difficulty in plotting orbits and making predictions on where celestial bodies should be became far more clear than the epicycle deferent equant system of Claudius Ptolemy. But Copernicus did use all of those same mathematical strategies that Ptolemy used, but also ran into the same difficulty in not being able to accurately predict where the celestial bodies should be. Each epicycle provided some comfort to solving the problem, but was never completely ideal. But when the sun was placed in the center, then the experimental evidences became so much more clear and corroborated, and predictability could happen. We're at the same position today in the cancer field as we were back then. We have thought of cancer as a genetic disease based primarily on a silent assumption and confirmation bias. It's a funny thing. When Watson and Crick discovered the structure of the DNA, it set off a phenomenal run to better understand biology at a molecular reductionist level. And everybody thought that we should be studying DNA in every which way. And of course, that led to tremendous advances in our understanding of biology for sure. And cancer was sucked into this whole whirlpool just like all the other fields of many of the other fields of biology, cell biology, biochemistry. Everything now could be explained in the form of genes. Um, and when cancer was looked at, of course, you could see broken chromosomes, you could see genetic mutations, and there was a conscious effort on the part of the National Cancer Institute, the National Institutes of Health, that we should um, focus more on cancer's genetic problems as a way to potentially solve the disorder. And in the 1970s and 80s, we launched the Cancer Genome Project, which was designed to find every type of mutation in tumor cells and thereby target those mutations that were supposed to be responsible for dysregulated cell growth. And that essentially is what cancer is: cell division out of control, dysregulated cell growth caused by genetic mutations in the nuclear DNA, and they're called somatic mutations. So the cancer is the somatic mutation theory of cancer says that over time and age and things like this, carcinogens, radiation, all these things, they'll cause mutations in the nuclear DNA, and those mutations will cause a dysregulation of the signaling pathways in the cell, leading to dysregulated cell growth. So the promise, then we got into precision medicine. We found so many mutations now we could target them with precision, personalized therapy, precision targeting drugs. And you hear a lot about this today with the immunotherapies and this kind of thing. And the hope was that we would eventually, with our deep knowledge of the genetics of cancer, we would be able to solve the problem because we could target these various mutations, preventing the dysregulated cell growth by targeting the very mutation causing the dysregulated cell growth. Very logical, supported by massive evidence showing how many mutations there were in cancer cells. But as our technology became more and more sophisticated, we started to find tens of thousands and millions of genetic mutations in some of these cancers. And then if you take a cancer out of someone's tissue, regardless of breast, colon, lung, brain, and you separate out the cells, and then you grow up each cell individually and look at the gene mutations, they're all different. From each cell in the tumor has a different constellation of mutations, making it extremely difficult to really figure out what's going on. And what's more, scientists began to categorize, reclassify some of the mutations in tumor cells. So not all of them are bad. We had then termed the driver, only the so-called driver mutations were considered the really bad guys. So you'd have passenger mutations that just happen to be mutated for something that really doesn't do much. But then the driver mutations are the ones that we need to target to manage the disease. And this has led to these enormous genome projects and the National Institutes of Health spending billions of dollars, pharmaceutical companies building drugs to target mutations, CAR-T, immunotherapies, Keytruda, Opdivo, they're all trying to target different kinds of mutations. And this is where it is today.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
The problem is we continue to use poisonous chemicals and radiation and these extremely toxic therapies. They were supposed to be replaced by our understanding of the genetics of cancer. So once we could capture the information from the genome projects, we would no longer need to irradiate and treat patients with toxic poisons. The problem is this has not come to fruition because cancer is not a genetic disease. The theory under which this enormous enterprise was launched was incorrect. Warburg originally showed that cancer was a mitochondrial disorder. And what I have done is followed what Warburg said. I went back to what he said, why he was correct and why he was not correct. We have cleared up a lot of the mess associated with Warburg's original observations. Clearly, Warburg was right that cancer starts with a chronic disruption of energy within the organelle that generates energy through oxygen, oxidative phosphorylation, oxidative respiration. We breathe air. Our cells get energy from the air we breathe. Every breath we exhale, CO2 and water, those are the waste products of normal respiration. Cancer cells get damaged, yes, from carcinogens, yes, from radiation, intermittent hypoxia, a variety of oncogenic viruses, but all of those provocative agents, if you will, damage oxidative phosphorylation chronically in some person's body, in a particular population of cells. And in order to survive, the cancer cells become suffocated because they can't use oxygen effectively. So there's a protracted or extended period by which the cell starts to adapt to a fermentation metabolism, which is energy without oxygen. Now, this is the energy that all cells, all organisms used before oxygen came onto the atmosphere 2.5 billion years ago. We had all living organisms before oxygen. They were all fermenters. They all used a non-oxygen-based energy because there was no oxygen in the atmosphere. So when the relationship between one form of bacteria, cyanobacteria, they started producing oxygen through photosynthesis. And then we started to develop metazoans, multicellular organisms, which we already now we're living today as the result of these events. We breathe oxygen and we produce CO2. But in the early development, there was no oxygen. So the waste products were these fermentation waste products. Cancer cells fall back today on these ancient pathways of fermentation, just as they were their heirloom pathways. They exist today, but they've become dominant in the cancer cell. And we know that because the waste products are products of fermentation. That is lactic acid and succinic acid. That's clearly telling us that these guys are not using oxygen. They're using these ancient fermentation pathways. And the other thing, as I said, today, all these different cancers have thousands of mutations. Everybody is different from each other. Every cell in the tumor is fermenting. It's not using oxygen as a major source of energy. So that tells us clearly what's going on in these cancer cells. It says they're all fermenters. And then when you know that, that immediately tells you they can't use respiratory fuels like fatty acids and ketone bodies because you need a good mitochondria to do that. And all cancer cells that we have found have abnormalities in the number, structure, and function of their mitochondria, thereby being dependent on a fermentation metabolism. So the solution to the cancer problem, I want to say a solution and probably the solution, is simultaneously target the two fermentable fuels driving the dysregulated growth while transitioning the body off to fatty acids and ketones, allowing our normal cells to use what we evolved to use. But the tumor cells can't use those fuels because their machinery to use those fuels is defective. Very clear. It immediately throws the whole field into a tivy, because you're realizing that a multi-billion-dollar industry built up around an incorrect theory. And the result of that is 1,700 people a day in the United States are dying from cancer. That's 70 an hour. 70 an hour. As we're speaking, 70 people will be dead as the result of this, because it's an incorrect theory. On all the things we're doing to treat cancer patients are based on an incorrect theory or ancient or old ways improved a little bit, but not understanding what the nature of the real problem is. So why are we continuing to do this? It's because a silent assumption that cancer is a genetic disease and confirmation bias, because everybody was led to believe this, and they all continue to think that way. So until that changes, we're not going to change the death rate significantly in any meaningful way.
Hmm. I've heard that it takes about 14 years for new research to get implemented in clinical practice, with doctors implementing new protocols and things like that. How long have you been doing research, or how long, you mentioned Otto Warburg. I'm familiar a bit with his initial findings. When was that, his research? And then
That was
when did you start
in the
yours?
1920s. He did that in the 1920s. He's a very fantastic character. He has a very colorful history. He fought in the German Prussian army, a cavalry officer in the First World War. Einstein told him he should be in the lab working. He was from a background of highly educated and top leading scientists. His peer groups were all these heavy scientists. He was a very powerful quantitative biochemist. He did not leave Germany. He was half Jewish, and Germans had a solution for anybody that was full or half Jewish. And Hitler did not exterminate Warburg, because he understood that if Warburg knew more about cancer than anybody else in the world at that time, Hitler feared cancer because his mother died of breast cancer. So he figured, if I'm not going to execute the guy that could potentially solve the problem of cancer. So he was allowed to continue to do his research in Germany during the Holocaust. And he didn't like Hitler in any stretch of the imagination, but he wanted to just do his research and be left alone. He did receive the Nobel Prize in 1931 because he found what we call the respiratory enzyme, the respiratory protein in the mitochondria that allowed us to generate energy from oxygen. He never received a Nobel Prize for his work on cancer. Hitler forbade Germans from receiving Nobel Prizes. He was nominated, but several times, but only one time did he get it. After the war, he was very arrogant and strong about his work, but he was swept away with the Watson and Crick discovery of DNA. So they more or less said Warburg was passé. His ideas were no longer relevant. He was just ignored, and they all ran off chasing genes, where we are today. So what I did is I went back, because I did research here at Boston College in our Department of Biology, and I was seeing results that were more consistent with Warburg than they were with the somatic mutation theory. So I said, who is this guy Warburg? I mean, I knew his name, but I didn't know what he actually did. So then you have to go back and do a real deep dive on what he said. I had papers translated from German into English, and I started to evaluate his formulations and things like this, and looked at it. Where did he make mistakes? Where was he correct? So we then started to do experiments, and fortunately, a lot of others in the field were doing experiments that actually supported Warburg's theory without them even knowing that it did. So what I did, I saw those experiments, and I said, geez, these experiments are supporting what Otto Warburg is saying. You guys don't even know about it. So I put a lot of this together with the nuclear mitochondrial transfer experiments and all these kinds of things. And then I looked, and I found papers saying there's no mutations in some cancers. I said, wait a minute. How can you have a cancer where there's no mutation? That doesn't support your somatic mutation theory, but they're fermenters. Oh, okay. And then we found all these driver gene mutations now in normal tissues that never form cancer. How you explain that? And then the nuclear transfer experiments, we take the nucleus of a tumor cell with all the genetic mutations and put it in the cytoplasm of a normal cell. You have growth regulation. Even though you have all the mutations still there, the cells are not dysregulated. On the other hand, if I take the nucleus of a normal cell and put it into the cytoplasm of a tumor cell, you get dysregulated growth, because the cytoplasm with the mitochondria controlling the events. Work from Israel and Seyfried, elegant, beautiful experiments. So you put all that together, and all of a sudden you have clear, massively clear evidence. Cancer is a mitochondrial metabolic disorder driven by fermentation metabolism, cannot switch to respiratory fuels like fatty acids and ketone bodies. It's unbelievable. Yet I think it's so shocking that the field can't accept it because of the confirmation bias. They have been brainwashed into thinking that this is a genetic disease, And no way in hell are they going to realize that it's too disruptive to the entire industry. It's the academic industry. The NIH says it's a genetic disease. It says right on their website, cancer is a genetic disease. All the pharmaceutical companies building drugs based on cancer as a genetic disease. The hospitals are making large amounts of money treating people with radiation and these toxic chemotherapies based on cancer being a genetic disease. If it's not a genetic disease, then what are we going to do? We have to remove the fermentable fuels and transition the body over to non-fermentable fuels.
Yeah.
You see what I'm saying? It's
And
too dramatic.
Yeah, I'm curious if we can talk more about that. So these two fuel sources, I've heard you talk about glucose, glutamine, that fuel this fermentation process where cancer proliferates. Will you speak more as to, my understanding would be carbohydrate intake, eating like a high carb, high sugar diet would contribute to these glucose levels. You speak more to those driving factors.
Yeah, well, I mean, there's been a large number. We published the first report, at least in the mouse, where we showed using linear regression analysis that the higher the blood sugar in the blood of the mouse that had the tumor, the faster the tumor would grow. And the lower the sugar, the slower the tumor would grow. And that's when we said, we saw this over and over and over again. And we said, how do you explain this? That's when I saw Warburg said the tumors are dependent on glucose for growth because that drives this fermentation pathway in the cytoplasm. So we said, wow, this is important. And then other physicians and clinics started to look at blood sugar related to breast cancer, colon cancer, brain cancer, all these different cancers, lung cancer, and they were able to show the same thing. If a person has lung cancer, brain cancer, colon cancer, their survival was significantly related to how high the blood sugar was. So clearly, it tells us you got to lower blood sugar if you want to try to manage your tumor. Despite that, they said, "Oh no, sugar has nothing to do." I don't know how you explain it. I mean, you show all these data, and then you say, "No, sugar has nothing to do with it." Sugar is not a carcinogen, but it's a fuel for the dysregulated cell growth of the tumor. So that was known for many years, and I think it's widely accepted among most logical understanding scientists that glucose is a driving fuel for the tumor cell, maybe not for oncologists in the clinic, but certainly for scientists that understand cancer metabolism, for sure. The glutamine issue is an amino acid, okay? So let's go back to the glucose. Most of the foods we eat in one way or another are broken down. Not all of them are broken down to glucose, and it's metabolized through the 10-step Embden-Meyerhoff-Parnes pathway to pyruvate. Pyruvate is a 3-carbon molecule, and it's fully oxidized in the mitochondria with CO2 and water produced as the end products. That's what the respiration tells us. Now, the glutamine issue, people in my field of cancer metabolism, we all understand that glutamine is another fuel that cancer cells can use. However, we have shown uniquely from my work here at BC and my collaboration with Christos Xenopoulos at Semmelweis University in Budapest, we have shown that the glutamine is fermented. It's an amino acid fermentation in the mitochondria. So this same organelle that produces the majority of energy through oxidative phosphorylation also has a capacity to ferment an amino acid with minimal use of oxygen. That's the big breakthrough. So Warburg did not know about this, and most cancer biologists don't know about it. And when we try to explain it to them, it's not easy to understand. So the field has used oxygen consumption as a surrogate for oxidative phosphorylation. So when you see a normal cell and a tumor cell, they're both consuming oxygen. So that leads individuals at a similar level sometimes. So that leaves some people to say, well, look, the cancer cell can respire just as well as the normal cell. The normal cell is using the oxygen for energy through oxidative phosphorylation. The cancer cell is using oxygen to produce reactive oxygen species, ROS, which are carcinogenic and mutagenic, causing the mutations in the nucleus. They're fermenting the glutamine. So if you're looking at the mitochondria, you see ATP energy coming out. But the normal cell, the ATP is coming out from an oxidative phosphorylation mechanism, where in the cancer cell, it's coming out from a fermentation mechanism.
Mm-hmm.
Even
Okay.
myself, I sometimes but we have the evidence to support that.
Wow. Yeah. So kind of going back to a little bit of like the basics of energy, like a healthy cell has different mechanisms that it can use. It can to make ATP energy, right? But
Yeah.
with when you have no oxygen, you have the anaerobic, correct? Like could you
No.
describe that a little bit
Yeah.
more for people? Yeah.
Yeah. So fermentation is energy without oxygen, usually. Okay. Let me give you an example. Heart attack. If people have a heart stops beating. Okay. And if you don't get the heart to pump the blood and you're going to be dead. All right. So it tells us that we need oxygen to be alive. Okay. That's a heart attack. Now, when you have a heart attack, immediately you see the waste products of fermentation appear in the bloodstream. So our bodies can survive for a short period of time, up to 5 or 7 minutes, depending upon what kind of shape you're in, without oxygen. We immediately shift to a fermentation metabolism, but it's very inefficient. So the waste products are lactic acid and succinic acid. And that's the waste products of glucose fermentation and amino acid fermentation, glutamine fermentation. So they immediately jump up in your bloodstream in order for us to keep ourselves alive. Now, if you don't have oxygen coming in a certain period of time, those fermentation pathways are not capable of providing long-term support. Now, cancer, let's look at the cancer cell. And the other thing, too, the poison cyanide, right? You know about cyanide? It's poison, right? Remember
Yes.
Jonestown? They put the cyanide in the Kool-Aid, and they always say the guy drinks the Kool-Aid, dies instantly. Okay, that shuts down oxidative phosphorylation quickly, and you die. It's a permanent shutdown of the Warburg enzyme, the Warburg cytochrome C. Very interesting. Cancer cells don't die from cyanide. Oh, it's great, right? So if someone were to have a big tumor in their body and say, I don't want to live anymore, let me drink cyanide, they'd be dead, but their tumor would still be alive because it doesn't die. It uses a fermentation mechanism. Also, in the presence, even in your body, when the tumor is growing, even in 100% oxygen, the tumor is still fermenting, still throwing out lactic acid and succinic acid. As I said, when you have a heart attack and you get oxygen, succinic acid and lactic acid go away because you're respiring again. The cancer cell cannot respire. It's locked in. So it's always throwing out these waste products. Those are signals. It's telling us these guys can't respire. What are they doing to survive? Glucose and glutamine fermentation. How do you kill the cancer cell? Take away its fuel, it dies, can't have no more energy. Not that complicated, right?
Right. So can you talk about how, because our healthy cells can also use ketones as energy. And so can you describe how being on like with this metabolic theory, like being on a ketogenic diet can help the healthy cells compete with the cancer cells and then shrinking and killing the cancer in a way,
Yeah,
or starving it?
Well, we started the ketogenic diet only because what really works is water-only fasting. And I said, if I'm going to come out and tell people that in order to get rid of your cancer, you can't eat anything for 30 days, they're going to say, well, this guy's some sort of a nut. So I worked in the epilepsy field for years, and we were using ketogenic diets to manage seizures in children. And was developed by Wilder at Mayo Clinic in 1921. It was very effective. And what you're doing is you're transitioning away from sugar, glucose, basically, to a new fuel, ketone bodies. And we can do that. Now, during our Paleolithic period, our species for hundreds of thousands of years was always in a state of nutritional ketosis, mainly because we didn't have highly processed carbohydrates, and we had a lot of exercise, and we didn't have Dunkin' Donuts on every corner. We had to kill and eat animals, anything, hunter-gatherers, a lot of exercise, a lot of meat eating, a lot of this kind of stuff. But all of it is that it lowers your blood sugar is still very low. You're not taking in anything, and you're exercising a lot. So you're using fat as a fuel, and the fat makes ketone bodies. So it's like a wood chipper. If you take a long chain fatty acid that's stored in your adipose tissue and you don't eat, your brain needs where your brain going to get energy if you're not eating anything? So you take the fat from your fat cells, your adipocytes, they go into the bloodstream and they go to your liver, And the liver, it's like putting a big branch into a wood chipper. So you get these water-soluble ketone bodies that go into your bloodstream, and they fuel the brain when glucose is deficient. They fuel kidney, they fuel muscle, they fuel. So all of our cells can, most of our cells can switch back over to ketone bodies or fatty acids, both, because those give us the energy when we're not eating food, okay? The tumor cells, unfortunately, the poor tumor cells, can't make that adaptation because you need a good, strong mitochondria, and they have defective mitochondria. So they can't switch to fatty acids or ketones, but our normal bodies can. So now, ketogenic diet, we just said, oh, well, that seems to be a little bit potentially easier to do than saying not eating food for 30 days, which is really hard. So it's not so much the ketogenic diet, it's the entrance into nutritional ketosis, the way we were during Paleolithic periods. So we developed the glucose ketone index calculator, which you can measure with a drop of blood. We call it the GKI. And the GKI will tell us whether we're in nutritional ketosis by measuring the ratio of glucose to ketone bodies. And if you can get a 1-to-1 on a millimolar ratio, then you're going to be in a state of nutritional ketosis. So when you're in that zone, what little glucose is available is going to the brain and the muscle. It ain't going anywhere else for the most part. So all the other organs, and they're going to be burning fatty acids and ketones. So the tumor cells in a state of nutritional keto, whether it's a ketogenic diet, water-only fasting, pescatarian, carnivore, doesn't make any difference. If you can get into ketosis, then the tumor cells are going to be at a competitive disadvantage. So they're going to be struggling for their existence, while the rest of the cells, which evolved to use this, are now you can survive, become healthy, then the tumor cells gradually to get degraded. But then you have to come in with the glutamine targeting to polish the remainders off. So it's you put the chokehold on the glucose, and then you come in with pulsing the glutamine, and it's a magnificent, non-toxic, therapeutic strategy to keep your body super healthy while you're degrading your tumor without toxicity.
Yeah.
How does that sound?
Yeah, that sounds amazing. I
good.
really want to get into the specifics with the pulsing, blocking the glutamine. But first, can we focus a little bit more on ketosis? Because I use a lot of the ketogenic diet, and I think it's helpful for people to understand why do we want to test blood ketones, and then and what's the difference, I guess, the benefits to tracking the GKI versus just ketone levels. I'll stop there.
Well, the GK, because that's the ratio. Because if you're now in diabetes, sometimes you can have high ketones and high glucose. We're talking about normal physiology here.
Yeah.
Okay, so the lower the blood sugar, it's because there's insulin responds to sugar. Okay, when you eat sugar, insulin goes up. All right, because insulin drives the sugar into the cells. This is the way it does. But if you don't eat anything and blood sugar gradually goes down, insulin goes down. But where's the energy come? So then there's another hormone called glucagon, and that turns on, and that makes gluconeogenesis and also mobilized fat. But the amount of sugar from gluconeogenesis, where we take amino acids and glycerol backbones, body can make sugar, but it's very little. And what little you have is going to go to your brain and your muscle, ain't going to the tumor. So, but what you're doing is you're transitioning your body away from a surfeit of sugar to using fats and ketone bodies as an alternative energy to sugar. And that requires a metabolic adaptation to this new condition. And there's a lot of ways people can get into this state. You can do water-only fasting. So we found that water-only fasting is very hard. And you know why it's so hard? Because our brains are addicted to glucose. So jumping off food, it's very hard. You not eat for 2 or 3 days, all of a sudden, you're ready to gnaw on something, but if you go slow, like you can take foods that have very, very little carbohydrate in them. You do exercise, you can eat meat or whatever would have low sugar. Your body slowly adapts. And then if you jump into water-only fasting, it's not so dramatic. But if you try to go cold turkey, it's very, very hard. So, and then we measure the ratio with the blood meter, right? Now,
Mhm.
the Libras are coming out. There's a free Libra for glucose, but it's not there yet for ketone. So eventually we'll have an arm patch that'll tell us
Cool.
what to do, and then
Cool.
we'll know what when they're in that zone. When we're in that zone, then you know this is the zone of metabolic nutritional ketosis, and you get healthy.
Yeah,
Yeah.
I love that. Yeah, and we want to use what's circulating in the bloodstream. We want to be testing what's in the bloodstream, not the ketone bodies that's leaving the body, right? To make sure that we're in
Yeah, well, yeah,
ketogenic
well,
state.
there are those machines or that you can blow into the thing and the light comes on for ketosis, and you can have urine strips, this kind of thing. But the blood is more accurate. It really tells you what's bathing your tissues, because the urine is those are releasing mechanism. I'm blowing out ketone bodies, which is acetone. Acetone is a non-enzymatic degradation product of ketone body synthesis. So you get acetone, you blow, and all of a sudden you get some light. And then you pee on a stick, and you have a little number there. But color change, you got a lot of ketones. But you can't. The GKI is a millimolar ratio of these two metabolites in your blood at that moment. And for the cancer patients, when they get into we put them in, we gradually transition them to a glucose ketone index of 2.0 or below. This is the zone that the tumor cells now start to suffer. And then when when the patient is in that zone, we come in now with the glutamine targeting pulsing to polish off these tumor cells. So it's a strategy, a diet and drug strategy. And I don't want to say that chemo and radiation and all these other things can't be effective. They're massively more effective when the body is in nutritional ketosis. So a lot of these radiation chemo, you don't need as much, you don't need to do it as long, and all of a sudden you can kill these tumor cells, minimizing toxicity. But you have to get the body first into nutritional ketosis, which we have defined as 2.0 or below. And I've had people struggle. Oh, I can't get to two. I said, well, you might have to do water only fasting.
Mm.
Sorry to say,
Yeah.
sorry to say, but we try to make it an easier transition, and
Yeah.
then you can put it on your cell phone, right? You got your cell phone here, so you can then monitor your GKI during a daily, time. We do use urine strips in between because the ketone strips are a little bit more expensive than the glucose strips. So these are blood. You take your drop of blood, you put the strip, you
Yep.
put it into the meter. Bing, tells you what your and then you take another strip, squeeze a little bit more blood, bing, and you get your
Yeah.
ketone, push the button, bang, here's your GKI.
Yeah,
Yeah.
with the
And
keto mojo, right?
Yeah, Keto Mojo.
yeah.
Yeah, that's a good one. That's already geared up to push the button to give you, it's a very simple division. You just have to divide glucose by 18, and then you get glucose in millimolar and divide it by ketone, which comes in millimolar. But no, too difficult for people. They just want to push the button, get the number. So that Keto Mojo has that. You don't have to really do much. Just push the button, get your GKI.
Yeah, and do you have a suggested time when people take their glucose and ketone levels before meals, after meals, morning, evening?
Well, that's the excitement of using your body as your own experimental machine. Everybody's different. So in order to know that, you have to take it several times during the day. At first, it's like experimenting with yourself. Eat certain things. You feel good. Okay, what's my GKI? It's still high. Okay, don't eat that. And then find out what time during the day. Usually it can vary. People sometimes get the lowest GKI in the late morning, sometimes in the middle of the afternoon. It can be all different times about this. So, it all depends on the person. So, and that's why I said once the person understands the concepts, then all of a sudden they'll know what their bodies and what's the time of day that they could be taking a secondary therapy. But it varies from one person to the next.
I get this a lot when I try to get people into ketosis, but a lot of people are afraid that the ketogenic diet isn't safe long term because we don't have any long-term studies. But my understanding, it's a metabolic state, right? So being in ketosis long term shouldn't be an issue. But what are your thoughts on that?
We were in ketosis for the majority of our existence as a species, right? We lived how old? We're about 1.5 million years as a species on the planet. So the Paleolithic period of our existence was always in ketosis because we didn't have any processed carbohydrates in our diet. Where are we going to get carbs? We're killing an elephant or a buffalo, chopping it up and eating it. We have a lot of energy there. Where's the carbs coming from this guy? The only carbs you're going to get perhaps is eating the liver. The liver is loaded with glycogen. But you're doing a lot of work to get that little bit of glucose out of and you have to eat a lot of it. You really have to pound it down. So we were always in a state of ketosis. The question now is, why is it so difficult for us to get back into that state that we evolved in? Hundreds of thousands of years, we're in one state. All of a sudden, within the last 50 years, we're in a totally different environment, preventing us from being in nutritional ketosis because the foods are so tasty, they're abundant, they're everywhere. And it's very hard. And you say, oh man, I do this, I do that. Yeah, because our environment is different. So as I said over and over again to a lot of people, I said our technology has evolved so much faster than our biology. We are all still Paleolithic people living in a new age. And we have an obesity epidemic because in order for us to survive as a species for hundreds of thousands of years, we had to be incredibly efficient in storing energy. Energy is stored in fat cells. So we are so geared for our existence as a starved species with very little, if any, carbs in the environment. Now we have all these carbs, and what do we do? Our biology is telling us, store the carbs. We don't know when we're going to be starving again. No, well, we got obesity, cancer, diabetes. This is evolution. People say, well, I don't understand evolution. I don't believe in it. Do you believe in the obesity epidemic? That's evolution in action. Do you believe in the diabetes epidemic? That's evolution in action. If you don't understand evolution, just look around. That's evolution. Thousands and thousands of years in a particular environment, all of a sudden shifted in a very short period of time, and our bodies are telling us this is not where we came from. This is not who we are. This is our technology and our environment, illustrating the biology our biology now interacts with the new environment. We have all these chronic diseases. Not complicated.
Yeah. Yeah. So when I have seen people be in long-term ketosis, their ketone levels, like the production, they can kind of drop over a period of time, like a year into it. Do you have any ideas about, should people cycle at that point of going in and out of ketosis to increase their therapeutic levels?
It's hard to say. Yeah, I know what you're saying, but my good friend Dominic D'Agostino, he's always in ketosis. He eats squirrels and stuff. I mean, he's like he's like the modern man living a Paleolithic lifestyle and stuff. And he's always in keto. He rarely, if ever, falls out of this. He's just living like we always were living. And so I think people make too much of this. So they feel they got to eat fat, they got to eat this, they got to eat that. What we find, though, it's a little bit more difficult to remain in ketosis on vegan or vegetarian kinds of things. But not impossible. But we evolved to eat meat. I mean, we made a lot of animals extinct by killing and eating them. Big paper came out of Israel not too long ago. Human beings killed and ate the strongest members of the animal societies, like not the very young or the old. They killed and ate the strongest ones because the marrow in the bones of these strong animals was better than the young and the old. So if you're eating the main protectors of your species, then the weak, the old, and the weak become food for the predators, and then the animals go extinct. So our species was responsible for the extinction of several animal species on the planet because we ate them, and we basically ate them, you know. And but you can get into ketosis with a pescatarian diet, a Mediterranean diet, a carnivore diet. Yes, vegan, but it's harder. It's harder. But I always use the GKI is the quantitative measure So you don't have to worry about what you're eating. People say, can I eat that? I don't know. You do it and tell me what what happens to your GKI. And they'll say, oh, it went up. Well, I said, don't eat that.
That's awesome. Yeah, I love it. It's a lot of people want to say, oh, this food is keto and this one's not. And it's just not true. A lot of people have blood sugar spikes from food sensitivities to certain foods, and that can impact your specific ketogenic diet if you want to do
well,
a
as I
ketogenic
said,
diet.
they're not using the GKI. They need to use the GKI. Then they know all these diet questions become less important. They're the ones who can know what they can eat, not eat. This guy might be able to eat what you can't eat. Who knows? Try it. Everybody walking around with it, if they're of this concern and they're worried about this and that, then they all should be knowing what their GKI is, so they know what their bodies are capable of taking in and being in ketosis. Not complicated.
One quick question too, with being in ketosis more long term, I know that being in a ketogenic state lowers the reactive oxidized species, the ROS in the cell, but that I've heard the ROS is also a signaling molecule, and so it can be somewhat needed. Is there an issue with getting too little ROS production on a ketogenic? Okay.
No, you're still going to get the adequate amount. It's having too much. It's having too much. ROS always come from, no, we will always have a small amount of reactive oxygen species, but burning ketones keeps it down so it doesn't get out of control. It's the ultimate health state. Nutritional ketosis is who we are. We're all in an we're bombarded to knock us out of the state every day on television. You'll see all these beautiful pizzas and donuts and Big Macs and all kinds of stuff all the time, right? And they're delicious. Don't get me wrong. I'm not one of these guys. I'll eat a pizza, jelly-filled donut. You kidding me? But I'm not going to do it all the time. And exercise is an extremely important component. So we have the cancer diet and lifestyle. If you want cancer, if you would like to get cancer, there are certain things you need to do. I'm not talking about preventing cancer. I'm talking about getting cancer. What is the best things we can do to get cancer? Minimal exercise. Don't exercise. Don't do whatever you can. Take the elevator. Never take the stairs. Always never walk anywhere. Get a ride from somebody. That's one way. Another way is you got to eat a lot of highly processed carbohydrate foods. You got to make sure that's the main component of your diet. God forbid you eat anything natural. So garden grown stuff, no, get away from that. You got to eat highly processed. If you want cancer, I'm telling you what you need to do, what people do. And if you look at what we do as a species, we're doing exactly what I'm telling you. You got to be under a lot of emotional stress. Got to have a lot of stress. You're freaked out all the time. Somebody says something on social media, on the cell phone, you get all bent out of shape. You don't exercise, you eat bad foods, you're under a lot of stress. Make sure you don't get a good night's sleep. Can't get good night's sleep. This is another thing. So you have bad sleep, bad food, no exercise, a lot of emotional stress, and you put all that together and you now are a prime candidate for getting cancer. And you do it over and over again, and you live in this kind of stressful, unexercised, poor diet lifestyle. And now we're seeing more and more young people. I get so many emails from people asking for help. I tell them what they need to do. I don't tell them, I'm not a physician. I cannot tell anybody what to do or what not to do. I just give them educational information and they make the decisions for themselves. We're seeing so many young people now. I'm telling you, in the 30s, early 40s, people with colorectal cancer, lung cancer, breast cancer. I don't. So clearly, we're having an earlier and earlier onset of cancer, which used to be mostly an old folks kind of a thing. Now it's like striking, and that's clearly related to our diet lifestyle. All those things that I said we're doing. There's another one here. Sleep. Yeah, a lot of emotion. Oh, the other thing is lack of happiness. Well, you're not happy about life. You're kind of always miserable or somebody. So you put all these things together that I just said, and it really is the diet and lifestyle for getting cancer. And unfortunately, this is happening. It's not only happening in the United States, it's happening all over the world. It's our globalization of food and technology has now put us at risk for not only cancer, but diabetes, cardiovascular disease, dementia, all of the different chronic diseases that we have.
Yeah. And I'm wondering if we could talk a bit more for the cancer patient of how to address the glutamine. So I've heard you talk about drugs that target glutamine. I'm wondering if you could speak to that. And I'm also wondering, are there physicians who do work from this orientation with the ketogenic diet and
You're
prescribing drugs that will
welcome.
address the glutamine as well?
Well, unfortunately, that's the problem. You see, we work under a standard of care, and the cancer standard of care is mostly radiation and chemo and surgical. Metabolic therapy does use surgery, ketogenic metabolic therapy, putting the body into a state of nutritional ketosis and then targeting the glutamine. There's no place anywhere on the planet that I know of now that's doing what we have recommended specifically. And the reason for that is they think it's a genetic disease and they're locked into a standard of care that says we must surgically debulk this, do a surgical procedure or radiation or chemical right up front, really quick. No. That's not the right way to deal with this problem. You have to bring your body into a non-inflammatory state, which is nutritional ketosis. Tumor cells start to get hammered. The tumors become much more circumscribed. The surgeon can then make a conscious attention to say we can debulk this whole thing real quick. We would transition you over then to targeting glutamine with a pulse. We don't use toxic chemicals unless we can assure that the toxic chemical will be, in fact, non-toxic in killing the tumor cells because we are now in a state of nutritional ketosis. So we have to reevaluate the whole standard, and we're not allowed to do that. The doctors who would love to do metabolic therapy to manage their patients, the system does not allow that. If they were to treat patients outside of the standard of care, their license to practice medicine could be in jeopardy. So clinics are not available. All of what I have said is what we know to do this effectively, manage this, and eventually will become the standard of care. It just takes time for the confirmation bias and the silent assumptions to crumble. And then there'll be a stampede to do this. But it's going to be requiring physicians to be retrained. They have to know how to do this. They're practicing now their art of medicine. It's unbelievable. They're going to be so excited because each person is going to be a different entity for them. It's not one shoe fits all. So like we do, we bring the people who can do this in these small clinics. Will look at the patients. They get their blood work all back. A lot of patients are sick from parasites, from obesity, from have all kinds of problems, diabetes. You got to bring that patient back into a stable state of health in part. Then you come in with the glutamine targeting drugs. The most effective one was 6-deoxynorlucine, which is done, which was used in the clinic on children and other cancer patients in the past. They were using it at an incorrect dosage and never targeted glucose at the same time, never brought the patient into a ketonic. When we bring we have preclinical models of the best of metastatic cancer and brain cancer and this kind of thing. When we get those animal preclinical systems into nutritional ketosis, we use small amounts of these glutamine targeting drugs. And you have to do it strategically because our immune system and gut and urea cycles all use that same molecule. So the tumor cells and our normal cells are using the same, our immune cells that will eventually pick up the dead corpses of cancer cells. They also need the glutamine. So if you're too aggressive on this, you kill tumor cells, but you paralyze the very cells needed to remove the corpses from the tissue. Mm-hmm. So you have to strategically know the biology of what you're doing and why you're doing what you're doing. You don't want to harm your gut. You don't want to harm your immune system. You want your gut to be healthy and your immune system to be strong. Because what we found out when we take away glucose and glutamine, tumor cells die. The immune cells can switch to ketone bodies, and they just become slightly paralyzed from the glutamine target, but they don't die. So as soon as you pull the glutamine back, these guys are alive. Bang, they'll take out those tumor cells. So you have to know how to do this, dosage, timing, and scheduling of these different procedures. And all while the patient is getting healthier. This is not, the patient gets healthier and healthier as you do this. So a lot of times the diabetes goes away, the hypertension, the high blood pressure, all that stuff goes away along with the slow degrading of your tumor. So it's really beautiful. It's unbelievable. When it's done correctly, by the right people who know what to do and how to do it, and by a compliant patient. The patient themselves must know this. And we find that if the family, the patient or the family are not on board, forget it. Doesn't work. So you got to have a compliant patient. And we find the ones that are most compliant, understand the concepts, are the ones that do the best. It's unbelievable. They really do well.
So you've done specific studies on the dosing, the pulsing, the length of time, and which drugs to use, and yeah. Can you talk about which ones?
That's where the cutting. That's the cutting edge right now.
Oh, okay.
We have not yet perfected that, and we're learning all the time on how to do that. That's why this metabolic therapy cannot be done with large clinical trials yet, because we don't know enough about the dosage, timing, and scheduling of the diet drug combinations. But that's not going to take too long. The problem is it's very hard to get funding for that type. It's not sexy science. It's trial and error kind of science. What works best for this person and that. So, and when you do a clinical trial, you can't say one shoe fits all. And besides, we're using diet drug combinations that we pull off and put on as we see how the patient is responding to this. So the whole concept of how to do clinical trials has to be reevaluated. We can't do what we're doing with these toxic drugs they give you, the cancer patients.
Yeah. So can the average person find a practitioner today? You said that there are people who are doing this well, but when it is done well, it works. But especially with the drug piece, like which drugs, how do you find a person who can give you the right drugs to work on
well,
this?
that's the other problem too. The Don is not available. And they know that. I would say they said it didn't work and it was too toxic. So I went back and it didn't work because they didn't use it the right way. And its toxicity was nothing compared to some of the toxic drugs we give to these cancer patients today, cisplatin, vincristine. I mean, these are horrible drugs.
Yeah.
The Don never was as toxic as these things.
Can you say that one again? I'm not, Ladon.
Well, the drugs that we use, like cisplatin, and we call one of them is Red Devil, doxorubicin. I mean, hair falls out. I always said when you see a bald cancer patient, you knew he was treated by someone who doesn't really understand the biology of the problem. Cancer cells do not hide in your hair follicles. So why your hair falls out? You're trying to kill a cancer cell, not go bald, right? Yeah. Well, they're just trying to poison everything and hope that's what we call medieval. You're using medieval. You don't have an understanding of the biology. How do you expect to know if this is going to work? And thereby, the grace of God, we get a lot of people surviving cancer, despite the fact that they were surgically mutilated, poisoned, and irradiated. I mean, you can think about it. The nice thing about metabolic therapy is that not only is it a good way to manage an aggressive tumor, but it's also can be used to prevent cancer. So part of it, obviously, we're not going to use glutamine targeting drugs to prevent cancer, but we can use a large part of metabolic therapy to prevent cancer. But when we want to manage cancer, we just bring in glutamine targeting drugs along with so I don't know anyone, maybe you guys do, I don't know anyone who goes down to the top cancer clinics, Dana-Farber, MD Anderson, Sloan Kettering, Moffitt, and all these. Please give me radiation and chemo as a prophylactic to get. Nobody does that, right? Why would you do it if you have cancer?
Exactly.
Well,
makes no sense.
Professor Seyfried, we're getting to the end of the time here. I wish we could talk more. But if people are interested in learning more about your work, supporting your work, your research, where's a good place to direct them?
Yeah, well, we get our support mostly from philanthropic contributions to Travis Christofferson's Foundation for Cancer Metabolic Therapies, and also directly through my university, Boston College. That money comes to our biology department and supports our research. And I know I said so many things, but we have it all published on open access papers in the scientific literature. So with Google, anybody who would have access to the internet can just put my name in and say, what is this guy publishing? and then you'll see the papers. Then it goes into a much greater level of detail. Our press pulse therapeutic strategy, which is kind of what I mentioned here, is given in great, it's the outline, the framework. The actual fleshing it out, we have a big paper under review right now, which is a treatment protocol that will be adapted into the clinics once it's finished and out there, which is really fleshing out the framework of what we're saying. So, and all cancers are responders. People say, "Oh, what about prostate? What about?" They're all the same. We can't find any cancer not dependent on glucose and glutamine. So they're all the same in that regard. They're all genetically different. They're all similar metabolically. So we know it'll work for all the different blood cancers, all these different people say, "Oh, it'll work on my cancer." I've looked at all the major cancers. They all have the same problem. They all have the same. They're all fermenters. So it's unbelievable, isn't it? It's unbelievable. So when people give us significant amounts of money, what I do is I include them as in our acknowledgments of the paper. So when people look at acknowledgments, like who's supporting our research, different people and foundations, sometimes that's all they want. Some guy says, "Oh, I just want to be part of this big change. I like to support it." And if he gives us the money, I put his name, support it. So the guy feels good. I say, "Well, look at my name's on this big paper that came out." So as in the acknowledgement section, we thank those guys that are supporting. It's going to be the new standard of care at some point in the future. So, but right now it's still at the very, very beginning stage. So you'll see when you're my age, everybody will be treated by ketogenic metabolic therapy.